Hello, my name is Stephen Munson. In this video I will be talking to you about an experiment that I've been conducting in the greenhouses for the last semester. In this experiment I am comparing competition between a native grass and an invasive forb at different levels of available soil nitrogen. There will be numbered citations throughout this video. The full citations can be viewed at the last minute of the video. The invasive forb that was used in this experiment is spotted knapweed. Spotted knapweed is a small flowering plant, usually around 3 feet tall when mature. It has purple flowers and distinct bracts at the base of the flower that give it the appearance of having spots. Unfortunately, I do not have any of my own pictures of an adult spotted knapweed plant, so I have provided one from the internet as a reference instead. Spotted knapweed has invaded a large portion of the United States, covering about 4 million hectares in 25 states as of 2004. However, this could be higher because of outdated estimates as well as underreporting. The map on the screen shows the states that spotted knapweed is currently considered to be invasive in. As I stated on the last screen, spotted knapweed currently covers 4 million hectares of land in the United States. That is equivalent to 15,444 square miles. To put that into perspective, the Upper Peninsula of Michigan has an area of 16,452 square miles. Imagine if the entirety of the Northern Peninsula was covered by a giant monoculture patch of spotted knapweed, with the exception of half of Luce County. One major reason why spotted knapweed has been able to invade such a large area of the United States is that it is allelopathic. This means that it produces a compound which inhibits the growth of other plants nearby it. In the case of spotted knapweed, the allelopathic compound that is produced is catechin. Catechin is released by the roots of spotted knapweed into the soil. In addition, spotted knapweed has a very high seed output. Each plant is capable of producing over 1,000 seeds. The seeds can remain viable in the soil for up to 8 years. This makes it difficult to remove spotted knapweed once it's become established in an area. Spotted knapweed also uses a large amount of nitrogen from the soil. This can allow it to competitively exclude other plants. These three factors have played a large role in allowing spotted knapweed to invade the United States. The area that is typically invaded by spotted knapweed is grasslands. When spotted knapweed has invaded an area, it can have a variety of impacts. One of these impacts is that it can increase soil erosion and runoff. This is because spotted knapweed has a taproot which does not hold together soil as well as the fibrous root systems of the grasses that are replaced by spotted knapweed. As previously stated, spotted knapweed has been shown to reduce the amount of available nitrogen in the soil. Spotted knapweed is also not as palatable to livestock as grass. It also forms monoculture patches that exclude any other plants. These four things all equate to degraded soil health and quality as well as a degraded ecosystem. Indian grass is a warm season grass that is native to Michigan. Warm season grasses tend to be more resistant to spotted knapweed invasion than cool season grasses, which is why Indian grass was chosen as the other plant in this experiment. Indian grass is widely distributed through the United States. Like spotted knapweed, Indian grass is known to remove a large amount of available nitrogen from the soil. The goal of the experiment that I will be discussing is to examine how competition between spotted knapweed and Indian grass is altered at different levels of available soil nitrogen. This experiment was set up as a factorial design. A factorial design means that it has more than one treatment and each treatment has more than one level, and every combination of treatments is used. For this experiment, the first treatment was the amount of available nitrogen in the soil. There are 3 levels to this treatment. The first level is normal soil without any alteration to the amount of nitrogen. The second was nitrogen added in the form of ammonium nitrate. The last level is soil that was amended with sawdust. The purpose of the sawdust is to encourage microbial development and uptake of available nitrogen, decreasing the amount of nitrogen available to plants. The second treatment was different seedings. This also has 3 levels. The first was spotted knapweed only, the second was Indian grass only, and the third was a competition pot with both spotted knapweed and Indian grass. The seeding treatments were crossed with the nitrogen treatments to produce 9 different treatment combinations. These 9 treatment combinations were repeated for 5 blocks to create a total of 45 pots. Another 5 pots were left unseeded with normal soil to act as blanks. An identification number was used for each pot, with the first number representing the block, the second representing the nitrogen treatment, and the third representing the seeding. For example, pot number 123 would be in block 1 with high nitrogen and would be a competition pot. The experiment is currently being conducted in Research Bay number 2 of the Barbara Kinchey Greenhouse at the Kinchey Hall of Science at Grand Valley State University. The Indian grass seeds that were used were collected from a population established using seeds from the Michigan Wildflower Farm in Portland, Michigan. The spotted knapweed seeds were collected from the Bass River Recreation Area in Ottawa County, Michigan. The soil that is used in this experiment is a sandy loam that was collected from the Bass River Recreation Area at the same time as the spotted knapweed seeds. The plastic flower pots that were used were provided by the Barbara Kinchey Greenhouse. The volume of the pots was calculated to be around 225 cubic centimeters. This was important for estimating the amount of soil amendments needed per pot. The sawdust was made from a block of oak using a chainsaw. It was dried in an oven to remove excess moisture. The ammonium nitrate was provided by the biology department of Grand Valley State University. The soil was sifted to remove any large debris. It was then mixed on a tarp to improve consistency between pots. On the right you can see a close-up picture of the soil. For the pots that required low nitrogen, roughly 4.5 grams of sawdust was added. This is equivalent to 3 kilograms per meter squared, a rate which was adapted from work by Everett et al. 2004. About 0.075 grams of ammonium nitrate was used for each of the high nitrogen pots. Each pot had a piece of filter paper at the bottom to prevent the sandy soil from leaking out. The weight of the pot and filter paper was recorded before the soil was added. And a spare pot was used to scoop about the same amount of soil into plastic bags labeled with the ID numbers of the pots. The amendments were added to the appropriate bags and mixed with the soil. The contents of the bags were then poured into their pots. The pots were weighed with the soil, but they were too heavy for the smaller scales, so I had to use a larger one. At this point, I had the weight of each empty pot, the weight of the amendments, and the final weight of the pots. From this, I could calculate the weight of soil in each pot. This will be useful for comparing grams of amendments added to grams of soil as a potential factor for the results. The Indian grass seeds were stratified in plastic bags with wet paper towels at 5 degrees Celsius. Spotted knapweed was stratified in the same way as the Indian grass, but was left in the seed pod. After stratification, the spotted knapweed seeds were removed from the seed pods. An intact seed pod can be seen on the screen. Each seed pod typically had around 3 to 5 seeds inside. The seeds were removed from the seed pods using forceps and a dissecting microscope. 300 seeds were needed initially, 107 of which can be seen in the picture on the screen. Each of the single species pots had 10 seeds, and the competition pots had 10 seeds from each. Here are some pictures of moving the pots from the lab where they were set up to the greenhouse. Randomization was used while setting up the experiment to help eliminate any accidental trends. The random number table that was used was from the book 1 Million Random Digits with 100,000 Normal Deviates by the RAND Corporation. The procedures that were randomized include scooping the soil, weighing the amendments, adding the seeds to the soil, and where the pots were positioned in the greenhouse. Here is a picture a few weeks into the experiment. A few of the pots had low germination rates and it was necessary to reseed them. The overall germination rate of spotted knapweed in the pots was around 20%, and the germination rate for the Indian grass was around 32%. The germination rates were used to determine how many seeds to add to each pot in order to get the desired number of plants. After reseeding, some pots had a significantly higher number of plants than others. This resulted in some treatments having a higher density of plants. As you can see in the table, pots with spotted knapweed had almost twice as many plants as pots with Indian grass. In order to prevent this from potentially influencing the results, some pots were thinned. The statistics program R was used to run a randomized block analysis of variance, or ANOVA, to make sure that there was no significant difference in proposed plant densities between treatments or combination of treatments. As you can see on this table, the proposed densities for each treatment are much closer together. The results of the ANOVA show that there was not a significant difference between treatments after thinning. The picture on the screen shows one pot before and after it was thinned. At the end of the experiment, the biomass of each species in each pot will be determined. Because the plants are growing in pots with sandy soil, it should be easy to retrieve both above and below ground biomass. The plants will then be dried in a 70°C oven until a constant weight is achieved. The nitrogen will then be extracted from the soil in each pot. This will be done using the potassium chloride method, which involves boiling the soil in 1 molar potassium chloride for an hour. After the nitrogen has been extracted, it will be analyzed by the Annis Water Resource Institute using an autoanalyzer. The results of the determination of plant biomass and available nitrogen in the soil will be compared using an analysis of covariance and the statistics program R. Currently, the spotted knapweed plants growing in the low nitrogen pots are showing signs of nitrogen deficiency. However, the Indian grass plants are also showing some signs of nitrogen deficiency in those pots. Statistics will be necessary to get a clearer picture of what role nitrogen plays in competition between spotted knapweed and Indian grass. If there is a difference, it could provide additional evidence towards nitrogen sequestration as a potential mechanism for warm season grasses' resistance to a spotted knapweed invasion. Thank you for watching this video. If you have any questions, feel free to contact me either in person or by email. My email is munsons@mail.gvsu.edu.